Hello everyone, my name is Shidharth Maheshwari and I will present my work REPUTE, which is an OpenCL based read mapping tool for embedded genomics. We are a group of researchers from Newcastle University and we are in collaboration with Indian Institute of Technology, Hyderabad. This slide presents the outline of my presentation. We will start with motivation, then I will present some background, then methodology, and then we will present our results. So, what is a genome? A genome is a complete genetic material of any organism. It is composed of DNA molecules starting from few thousands to billions of DNA molecules. Each DNA molecule is made up of 4 nucleotide bases, adenine, cytosine, thymine, and guanine, which can be represented as alphabets A, C, T, and G. So genome is basically a very long sequence of A, C, G, Ts arranged in a particular manner. What is genomics? In layman terms, genomics is the study of genome. And what are its applications? So genomics finds applications in many fields, few of them being biotechnology, agriculture, forensics, and medicine. Also in social sciences. So for example, we want to do a genetic diversity of a population to determine the effects of evolutionary process, for criminal casework, for identification of missing person, for creating artificial genomes, organisms, plant breeding. However, the most important and foremost application of genomics is medicine, which includes a research and development of more than 6,000 genetic disorders, including cancer, diabetes, arthritis, high blood pressure. It's also a front runner in undergoing transformation of medicine from reactive to predictive, preventive, participatory, and personalized form. This transformation is aimed at providing patient-tailored therapies to predict and prevent genetic diseases before symptoms appear, or in some cases even before birth. Many countries, including the UK and US, are planning to sequence very large proportions of their population and also include genomics in the routine test. So to set up infrastructure for routine genomics, we need to first identify what are the challenges in this field. So genomics is a big data contributor. We can see that since the advent of next generation sequencing from around 2006 to 2007, we have seen a steep rise in the amount of data being produced by genomics. It is predicted that in coming decade, it will surpass astronomy, YouTube, and Twitter. Both of these diagrams show the same trends. This plot presents how are our computational capabilities scaling with respect to the amount of data being produced by genomics. This blue line shows the Moore's law, this red line shows the historical growth rate of data, and this yellow line shows the Illumina estimates of growth of data by 2025. And we can see that in both the cases, the amount of data being produced will outperform the improvements in computational capability. It is known currently that up to 560 GB of data per human being is produced at sequencing stage. The processing done by genomics involves diverse range of approaches, and it may need to do the same kind of processing on the entire data again and again multiple times to derive more and more information. The estimates show that we will require about 10,000 trillion CPU hours for processing entire genomic data by 2025. State-of-the-art genomic facilities include supercomputer and high-performance servers. Improvements to CPU capabilities, as anticipated by Moore's law in the previous slide, should help close the gap, but trends in computing power are often geared towards floating point operations and do not necessarily provide improvements in genomic analysis. In genome analysis, it includes string operations, and memory management often poses the most significant challenge. Hence, we can conclude that there are 3 major challenges to widespread adoption of routine genomics. First one is high infrastructural setup cost. Second is massive amounts of data. And third is massive energy consumption. So what do we require? We require something which is low cost hardware. We want high performance and we also want low energy consumption. We believe that embedded genomics can provide all the 3 together. We will now proceed forward to the background. We'll discuss some previous works and then key contributions. So how is genomic data obtained? Genomic data is obtained using a process called whole genome sequencing, which has three stages: sequencing, genome assembly, and analysis. Sequencing takes place in dedicated sequencing machines, which involves several chemical reactions, which breaks the original genome of organism into smaller pieces and then sequences it in the form of constant length reads. These reads are then recombined together to obtain the original genome, which falls under this genome assembly stage. Both the genome assembly and analysis are the computational pipelines and are processed on CPUs. Here is a list of state-of-the-art read mappers used by bioinformaticians. As we can see, all of them have been tailored for CPU-based implementation and are oblivious to different hardware resources available underneath. Several tools have been proposed to accelerate read mapping on GPU, but did not receive widespread acceptability due to platform-specific implementations. Similar is the case with FPGA implementation. GPU and FPGA-based mappers often require platform-specific software implementation methods. In our experiments, we were successful in executing only Razer S3 and Hops 3 and Coral on embedded single-board computer. We can conclude saying that Coral and REPUTE are the mappers which are cross-platform and are implementable on heterogeneous platforms. Here we present our key contributions. REPUTE is an OpenCL-based read mapper for heterogeneous systems. It's the first implementation of its kind which demonstrates a computational pipeline on an embedded platform. We obtain high performance and low energy by employing hardware-software co-design approach. We obtain high performance by designing a low memory footprint kernel which employs a novel dynamic programming-based filtration methodology. Combined with OpenCL, we use this tool to implement it on a very low power platforms such as embedded platform and obtained extremely low energy compared to workstations. Our tool is Python and OpenCL based, which is often the most used language by data scientists and bioinformaticians. We demonstrates up to 13x performance gain and up to 27x energy savings on embedded platform compared to a general purpose computer. We will now present our methodology and our proposed dynamic programming based filtration method. Coming back to our previous slide, we will now discuss how read mapping is performed and how is genome assembled. We reassemble the genome using read mapping approach. Read mapping is very similar to solving a very large scale jigsaw puzzle in which we have an approximate idea of how our final picture should look like, and we reconstruct the final picture using small pieces. Similarly, we are already provided with a human reference genome, and then we locate those small reads in the human reference genome to reconstruct the original genome of an organism. The problem is that every human genome is about 99% similar but has 1% or less differences. These differences are accounted as errors in these reads, the red ones. We can see that read 1 matches in this location, but one of the bases do not match. Similarly, read 2 match in this location, but 2 of the bases do not match. These are the errors, and hence exact matching algorithms cannot be applied in read mapping directly. This makes it an approximate string matching problem. Now we will explain what are the sources of errors in reads. The first source of errors are natural variation, which can be due to differences in a species and or maybe differences within a species. The second one is due to the sequencing process. These errors have unknown distribution and can be found anywhere in the read and may differ in the number of errors as well. However, in general, we have an estimate from our sequencing process that errors may not be more than 5% of the total length of the read. These errors make exact matching impossible, and we will have to find ways to account for the errors while matching the read in the reference genome. Read mapping consists of 3 pipelines: preprocessing, filtering, and verification. We use reference genomes in 2 of these processes. We will start with discussing verification and we'll go backwards to filtering and preprocessing. We will do this in order to explain why our novel filtration methodology has given us high performance and leads to low energy consumption. In this slide, we will explain what verification is and how it is performed. So verification is a way to find out with what error does a particular read match to a particular section of the reference genome. We attempt to find out the number of errors with which it matches and the starting location of this read in the reference genome. This location of interest in the reference genome where a read may match within a certain error is called candidate location. We use a semi-global dynamic programming based algorithm, its variant, which is known as Myers bit vector algorithm, to perform verification. The complexity of Myers bit vector is O and M by W, where N is the length of one string, M is the length of another string, and W is the bit width. We know that reference genome is over 3 billion bases. Hence, it is almost impractical to apply verification with respect to every read in the entire human reference genome at one go. We can also, another approach can be used where a reference genome is divided into smaller sections and then we apply this verification with respect to each read in each of these sections. However, no matter how small a section we use, a dynamic programming based algorithm is in itself a time consuming and requires a lot of memory footprint. Secondly, each read may have originated from multiple locations, maybe from different locations rather than what we are finding, because human genome has almost 50% repeats section. The parts of those genomes repeat itself in several places. Hence, we cannot use verification in the entire reference genome. So what we want is the candidate locations where a particular read can be found. Fewer the number of candidate locations, faster will be the entire mapping process. Thus, our objective is to find the candidate locations first so that we can reduce the total number of verifications. This slide presents an overview of what filtration does. So filtration uses a read, applies a FM index backward search, which we will explain in a little while, on a data structure, pre-processed data structure obtained from the reference genome to obtain the candidate locations so that it can be verified using Myers bit vector semi-global dynamic programming variant. An efficient filtration algorithm attempts to minimize the total number of candidate locations for each read. We should remember that 560 GB of data, as we have mentioned before, is produced at the sequencing stage. So that means we have millions and millions of reads. So fewer the candidate locations per read, our entire mapping process will take less amount of time. FM index backward search is a very known search method which is applied on a FM index data structure. We will discuss this in our coming slides. But we should note that FM index backward search is an exact search method and hence cannot be applied directly on a error prone read. To account for the errors and also apply exact matching using FM index backward search method, which is one of the fastest method provided FM index data structure is available to us, is to use pigeonhole principle. Before delving into that, I would like to explain some terminology of definition of k-mers. So for example, we divide a read into small sections of length k. Those small sections will be referred to as k-mers. For example, this diagram illustrates 8 mers derived from a read. Now let us assume that we have a read of length 100 and we allow for an error of 5%, which is 5 errors. Now pigeonhole principle states that if we divide this read into n plus 1 errors, that is 6 sections, 6 k-mers, then one of those k-mers are bound to be error free. For example, over here we divide this read into 6 sections, and we can see that except for rest of the 5, one of these sections are error free. This is due to the pigeonhole principle. Now we have discussed that an efficient filtration algorithm is the one which minimizes the number of candidate locations for each k-mers, because wherever these k-mers will match, we will have to verify the entire read in those genomic positions to find whether there is a match within a given error or error value or not. So we can also see that first k-mer has about 120 candidate locations and of length 20. Similarly, this k-mer is of length 17, has 66 candidate locations. And similarly, the error-free k-mer is of length 21 and it has 15 candidate locations. We should remember that these k-mers are being searched as using FM index backward search. We are doing an exact searches on them in the reference genome, because we don't know where the error is not present in which of the k-mers. So we have to match all the k-mers, and we are sure that one of these k-mers will lead us to the original position in the reference genome where this read could have originated. We should also note these optical dividers in red lines, which demonstrate the boundaries of different k-mers, and an efficient algorithm will determine these optimal dividers so that the overall number of candidate locations for the entire read in the reference genome is minimized. This slide demonstrates the frequency of these with which these k-mers occur in the reference genome with respect to the length of the seeds. So suppose we focus on this image where 15 mers have been used, derived from the entire human reference genome, and they have been, and we have on the x-axis is the number of times any of these k-mers appear, and on the y-axis we can see the total number of k-mers. We can see that there are very few k-mers which have very large number of occurrences in the reference genome. That means if a read has such k-mers, if a filtration algorithm finds these k-mers, that will lead to a very large number of candidate locations and hence will increase the overall mapping process. On the other hand, if our k-mer selection process selects k-mer from this region, this will have very few number of candidate locations in the reference genome and hence will help us reduce the overall frequency or overall mapping time. As discussed previously, we need to find k-mers with the minimum number of candidate locations. We propose our algorithm, a dynamic drawing-based filtration method, to identify those k-mers which will lead to a minimum number of candidate locations in the reference genome. Let us assume that we have an error of 5, so delta represents the error, and S minimum is the minimum k-mer length which we are working with. Given delta is equal to 5, we will divide our read into 6 sections. Our read length is 100 over here, and we will identify the optimal dividers for each of those 6 k-mers so that our total number of minimum candidate locations can be minimized. To do that, we start with first 2 k-mers and then reserve the space for the rest of the 4 k-mers. The orange box over here is the minimum k-mer length for 2 k-mers, and the blue box over here reserves the space for rest of the 4 k-mers. Hence, we can see the middle part is the exploration space. So for each prefix, we will try to identify 2 k-mers such that the total number of candidate locations of those 2 k-mers is minimum. When with each iteration within this iteration, we keep on increasing the prefix size till here, and for each prefix, we find the best solution for 2 k-mers. After storing this value, we move on to the second iteration over here, in which we use those previous values and we find the optimal solution for overall 3 k-mers within up to this exploration space. These iterations are continued for delta times, which is 5 iterations, as the number of errors is 5 over here. And in the final stage, we find a solution for all the 6 k-mers such that the overall number of candidate locations for the entire read, including all the 6 k-mers, is minimized. After doing this process, we use backtracking to identify the optimal boundaries where each read starts and each k-mer starts and each k-mer ends. Then each of those k-mers are then searched in the reference genome using FM index backward search, and then they are verified. Here we demonstrate how we do pre-processing. We take the reference genome, we apply Burrows-Wheeler transform, and we obtain different matrices like tally matrix, suffix array, and F matrix, and we encode the reference genome as shown over here. FM index data structure is a very well-known technique, and anybody interested in that can find sufficient materials online to understand it. We will now move forward and present our results. We have considered two systems. One is Intel Core i7 CPU, which is a workstation with 4 cores, 16 GB RAM. It also consists of 2 NVIDIA GPUs. System 2 is a high-key 970 embedded SoC platform with 8 core ARM processors and 6 GB of RAM. We use 2 million real human reads of different lengths for evaluation and map it to chromosome 21. The error ranges are between 3 to 5%, and Razor S3 is used as a gold standard. We use Razor S3 as a gold standard because most of the previous algorithms have also used Razor S3 as a gold standard, as it is considered to be one of the most accurate read mapping. This slide presents results for the homogeneous scenario, which means that all the read mappers were implemented only on the CPU of the workstation. We can see that accuracy-wise, REPUTE performs either same or better than rest of the previously proposed read mappers. With respect to mapping times, we can see that we are faster than all the previously proposed read mapper, and we are faster by almost a factor of twice with respect to Coral, especially for longer reads and higher error values. This slide presents the results for a heterogeneous scenario in which we use the GPUs as well. We can see that only Coral and REPUTE were capable of using GPUs along with CPUs to distribute the workload among them. We can see that when we use REPUTE, when we use GPUs, our computation time again falls by almost a factor of 2. So we are almost up to 2x faster than the homogeneous scenario. This result demonstrates that with the availability of GPUs and other processing units on a heterogeneous platform, if they are used to distribute the workload, we can achieve higher performances. This slide presents the embedded scenario in which we have implemented 4 of these tools on high-key 970 platform. We can see that for read length of 100, REPUTE outperforms all the other read mappers, especially for lower error values. The accuracy is either same or better than the previous read mappers. For higher error reads, for higher length of longer read lengths, for low error values, again REPUTE outperforms all the previous read mappers. We produce the same value, or we are almost a little bit higher than HOPS 3 for higher error values. The diagram on the left shows performance gain due to distribution of workload on GPU. We can see that the computation times keeps on falling as we distribute more and more reads to the GPU. After one point, beyond one point, we can see that the time, again, overall computation time increase because the overall time taken by GPU to process those reads is more than the overall time taken by the CPUs. Over here, we can see that for different k-mer lengths, minimum k-mer lengths which we discussed, for different minimum k-mer lengths, the overall read mapping time reduces when we increase the k-mer length up to a particular level, beyond which it increases again. That means the filtration algorithm works better when there is at least some exploration space available to it. This slide presents power and energy consumption figures. Power was measured using a power meter right at the source. We can see that REPUTE outperforms all other read mappers with respect to the energy consumed in read mapping process. With respect to the workstation, embedded systems can save up to 27 times energy utilized in mapping process. This we attribute this savings in mapping, savings in energy compared to other read mappers to our dynamic programming based filtration method, which has significantly reduced the overall number of candidate locations, and hence it has reduced the overall mapping time. And secondly, we have also done many memory optimizations to design a low memory footprint kernel. Coming to conclusions, we have developed a normal dynamic programming based filtration method, which can give us up to 13x performance improvements. This is first of its kind implementation in embedded platform, showing 27x energy savings. We demonstrate the advantages of using heterogeneous hardware. We have used Python and PyOpenCL, which allows for faster prototyping and modification. Ours is a cross-platform tool and can be used on any OpenCL conformant device, and the source code is available as an open source on GitHub.